on pharmacoinformatics. It's a topic which Naipur Mohali started across the world first time and graduating many students, at least 20 every year at the master's level. This session we have 4 speakers, and I shall be Professor Rebecca Wade is the first speaker. She studied at the University of Oxford, and then she carried out research as a postdoctoral fellow at Houston as well as Illinois. And then she took up position at Structural and Computational Biology program at the European Molecular Biology Laboratory. So since then, since 1990s, we are aware of her research work. Professor Wade and myself bumped into many conferences in India as well as in Germany. We attended similar conferences on computer-aided drug design as well as computational biology and many other things. I had an opportunity to visit her laboratory 2 times already at the HITS Heidelberg. She has a collaboration with our Professor Sangamwar. A very interesting collaboration is in progress. And one of our bright students from our place, Dr. Prajwal Nandrekar, he went and visited her lab and contributed, and she liked Dr. Prajwal very much and his work. So thank you, Dr. Wade, for making it to come here and gracing this occasion. I request you to present your work.
Okay. Thank you very much for the kind invitation. Shall I use this one? Okay, thank you. Yes, and so thank you for the introduction, and thank you very much to the organizers for the invitation to come and speak here. I'm going to tell you a bit about what's going on in Heidelberg, at Heidelberg Institute of Theoretical Studies, where I have my research group, as well as University of Heidelberg. So I'm going to be talking about our simulation efforts to look at the interactions of drug molecules with polymer excipients, protein crowders, and target receptors. So these might seem quite disparate subjects, but I hope you'll see how they all connect by the end of my talk. Okay, I'm not quite sure where I have to point this, but it's not going forward. I've tried both, so it doesn't work either, sorry. Oh, now it worked. Okay, well, I'll just keep talking, I think. So the usual starting point for our work is a protein structure. We usually look at protein interactions that can be with drug molecules or other proteins. The structure can come from experimental data or increasingly from predictions. As you probably know, there's been huge advances in the application of deep learning approaches to predicting structures such that we can now have a much bigger array of potential protein structures that we can tackle and study with protein structure-based approaches. And one of the applications for studying these structures is structure-based drug design. And I'm going to start at some point when my slides go forward to give you an example of a recent structure-based drug discovery project that we've done, and then show it how it connects to dealing with these other topics. Oh, okay. So how do we tackle doing our calculations? Well, we're typically building physics-based models that allow us not only to look at structures, but to look at the dynamics of these molecules, which are critical to their function, and that can require doing calculations on supercomputers. But we aim also to combine this sort of modeling and these simulations with information that can be from evolutionary information, from sequence analysis, for example, or experimental data, and try to use these in a concerted way. And if the next slightly, oh, maybe there's something here. Here, here. Do I point it there? Where should I point it? In the middle of the screen. Okay, and so, yeah, we, as I mentioned, combining with experimental data, and of course, with human interaction is critical to all of this. And so we try to aim to do sort of a data-enhanced, multi-scale molecular simulation work. Multi-scale because the motions and the both in spatial and temporal terms of biomolecules are really cover many, many scales. So let's see. Now, in addition to thinking about how, say, a drug is going to interact with its target, if we're going to have an effective drug, we want to think about other properties too. And so we also we're basically trying to go for to develop a molecular level understanding of drug kinetics through molecular simulation. And so we are, for example, looking at how a compound might be, a compound that has to be orally available, how we can actually achieve that. And this is the work that's being done in collaboration with Abhay here on looking at how one can use polymers as excipients to make the active ingredients bioavailable. And I'll talk a bit later about molecular dynamic simulations to study this. But also, as you know, as a molecule gets close to its receptor, it has to pass through a crowded environment, a macromolecular crowded environment, the cellular environment. And so we've been asking, how does it diffuse through that, and how can this affect the whole kinetics, which can be affected by this process, getting to the binding site, the kinetics actually on the target, where we're also developing approaches to look at this. And so I'll talk a bit about what we've done to look at drug diffusion in crowded environments. And another important aspect is the drug metabolism. And here we are doing simulations of, to look at the mechanism of cytochrome P450s. So I'm not going to talk about that today, but I will come to these topics. But first I want to tell you a little bit about really targeting a receptor. And I'm going to give you an example of a recent work that we did aimed at tackling neglected tropical diseases that are caused by trypanosomid parasites. So these are diseases like Chagas disease, leishmaniasis, and sleeping sickness. I can't escape from the microphone. Okay. In order to tackle these parasites, we have targeted a particular enzymatic pathway, the folate pathway. So key enzymes in this, dihydrofolate reductase and thymolate synthase, and the well-known inhibitor is methotrexate, which is used as obviously as an anti-cancer agent. But noticeably in these trypanosomids, there's an enzyme called pteridine reductase 1 that can, when this enzyme, dihydrofolate reductase, is inhibited, can catalyze a bypass reaction, and so it can overcome this inhibition. So in this study, what we wanted to do is actually design an inhibitor for this enzyme, which you don't have an equivalent enzyme in humans, so it's a good target for having specific antiparasitics. But at the same time, we also want to inhibit the other parasitic enzymes without inhibiting the human ones. So if we think about, say, in the case of Leishmania, we might have the parasite here inside a human cell, and we want to target the pteridine reductase, the hydrofolate reductase, the thymolate synthase of the parasite, but we don't want to target the off-target human dihydrofolate reductase and thymolate synthase. And so we have the problem of basically a multi-target optimization. And so we basically put together a team of scientists in a European Union-funded project called NM-TRIPI for New Medicines for Trypanosomid Infections, which included people doing with various different experimental techniques as well as computational work. And in particular for the structure-based drug design, we had crystallographers, we did molecular docking, and we had enzyme assays. And we aimed to have inhibitors that were based on the folate scaffold, a sort of classical approach, as well as new scaffolds from virtual screening or fragment-based drug design, or from screening natural compound libraries or plant and fungal extracts. So I'm going to talk a bit about the work on the folate-like inhibitors. And crucial to all of this work from the structure-based perspective was really this mapping of these on and off targets so that we could identify parts of the compounds that we wanted to change to get selectivity. We developed a pipeline in order to do a systematic mapping of these on and off targets, including sequence alignment, structural alignment, analysis of all the interaction networks, and the conformational variability of the binding site. Here you see the superposition of crystal structures. You can see there's quite a lot of dynamics in this binding site, and we looked at this with our tool for exploring transient pockets in proteins. And so with all these different approaches, we could come up with basically a guide for distinguishing between the on and the off targets. And I just show you sort of the net result of this. So here is a folate-like molecule in the binding site, one of these binding sites, and we can divide it into sort of three parts of this molecule and look at the interactions in Trypanosome brucei and Leishmania enzymes, both the pteridine reductase and the dihydrofolate reductases, and then identify the key residues interacting, and where you see these little residue signs sort of at an angle, that's where we have a difference in the sequence to the human protein, so that helps us to get selectivity if we can target this region. You can see we have considerable variations, and although both these enzyme types bind to these folate-like compounds, it can be quite different in the way they do it. For example, here in the pteridine reductase, this tail end here is fully exposed, here it's enclosed in the dihydrofolate reductase, and between different species on this end you have quite a lot of differences. So what we did in the case of coming up with folate-like compounds was first to start with 8 compounds that had been characterized in the literature before, which were not specific for our main target being the trypanosomal PTR1. And then based on this mapping, we designed 20 new compounds that were tested iteratively in experiments And with that, we were actually able to come up with compounds that had much better selectivity for targets of interest and were very potent, approaching even apparent picomolar enzyme inhibition against this enzyme. But what we saw also was a problem that when we looked at the activity on the parasite, it was rather low and not really directly correlated with the activity on the enzyme target. So then based on this mapping, we did a sort of combinatorial analysis to come up with over 2,000 potential candidate molecules. And then we prioritized them based on considering not only the properties on the binding sites in the different enzymes, but also the physicochemical properties of the compounds. And these physicochemical properties we did a correlation analysis with the known activities of the compounds on the parasite to develop basically a model to tell us how we could optimize both for the target and for the parasite. And in particular, we saw that for getting antiparasitic activity, we needed to have a certain amount of hydrophobicity in the molecules. So by filtering through here, and I'm not going to go into details with these things, we came to 600 compounds. And from this, we selected just 6 compounds to actually test experimentally, to make and test. And you can see here, if we look at the activities against the parasite, this is in percentage inhibition here, so the compounds we started off with, which didn't have good activity at all. This was our initial set of compounds where we explored this. And this was our final set of 6 compounds. And you can see that 3 of them actually have really good activity here, and then the submicromolar, you see 50 level against the parasite, while actually still having very high affinity, or IC50s, against the target enzyme, and very good selectivity for the parasite enzymes with respect to the human dihydrofolate reductase, which is shown here. So these were our compounds here. We also checked the liabilities for cytochrome P450 metabolism, HAG, or mitochondrial toxicity and growth inhibition. And here you can see, compared to the reference compounds, our final compounds are not so good. So the more orange, the worse they get. Green up here is good, orange here is bad. So we can see that, in a further step of optimization, we should consider not only the binding to the many different targets and off targets that we're considering. We should also consider what's necessary for this molecule basically to get into the parasite, but also then these properties that are important when we consider that we want them to work for humans. So really what I wanted to show here is with a sort of classical approach with using virtual screening and docking, we can come up with very good binders and good inhibitors of our target in a way that we can get selectivity. But we do need to optimize other properties as well, and that's absolutely key to having good molecules in the end. And that really means we have to simultaneously deal with lots of really different types of data. So I've talked about we really need to know how, to get our molecule to a parasite, and that also means we need to know much more about the biology of such parasites in order to be able to really do that in a way that we understand. But of course, we also have to deal with the drugs going to humans. And so I'll approach that problem now by first considering simply what's going on when we consider drugs diffusing in cellular type environments. So basically, normally we think about, if we were thinking about in vitro experiments where we've got really dilute solution, but when we go to cell, we have very high concentrations of macromolecules and many metabolites as well. And we got interested in this based on work that Gideon Schreiber did at the Weizmann Institute, when he was looking at the catalytic efficiency of an enzyme, a beta-lactamase, and saw that it was reduced and variable in living cells. So basically, he had this marker substrate that he could follow, and if he looked at the catalytic efficiency in vivo versus, say, the recombinant protein, you can see that it just has a lower value and more distributed. And they did various experiments to try and work out why, and they proposed that this was because of slowed diffusion of the substrate in the in vivo environment. So if that's the case, then we'd also expect that this would be relevant for drug-like molecules, and so this is what we wanted to investigate. But in the cellular environment, this is very difficult to work out what's going on because of its high complexity and heterogeneity. So we decided then to look at simple systems that we could study in parallel by experiment and by simulation. And so basically the system is shown here. In blue, this is proteins, and in red, these are small molecules diffusing protein crowders. So we've got one type of small molecule and one type of protein crowder. In Gideon Schreiber's lab, about a day had worked on developing a method, a method called line FRAP, that enables one to measure the diffusion rates of such molecules. And what we did in my lab, this is work of Ariane Nunes Alves, a former postdoc, was molecular docking to look at the intermolecular interactions and Brownian dynamic simulations to compute translational diffusion rates, which we do using a method and software we developed for a number of years called SDA for simulation of diffusional association. So there is a movie here to show you one of these Brownian dynamics trajectories. So you can see that we have our small molecules diffusing and our proteins diffusing as well. And so we were aiming at a direct comparison of simulations and experiment. Now, for this study, we had to select molecules that we could study that were accessible to the FRAP experiments. So they needed to have suitable fluorescent properties. And so we selected these 4 compounds. So fluorescein is a biological marker, and 3 drugs, doxorubicin, a kinase inhibitor, and quinacrine. They have quite different properties in terms of charge, in terms of hydrophobicity, and so on. And then we took 3 protein crowders, so myoglobin, henegrate lysozyme, and bovine serum albumin, and looked at their effects separately. So now, what turned out from this, I shall tell you the conclusions now because they are quite complex, is that we did see that both protein crowders and the surface, these experiments are done with a liquid drop on glass, so the glass surface basically can affect the translational diffusion of these drug-like molecules. And in fact, we saw that this was really different in each of the 4 molecules we looked at. So I'll just walk you through this. In the case of quinacrine, in fact, it was totally unaffected by the addition of protein crowders or surface interactions. On the other hand, fluorescein, similarly small molecule, was slowed by the protein crowders, especially by surface-absorbed proteins. So basically, when we added protein crowders, what happened is that the protein crowders could interact with the surface and the molecules bound to the proteins, and then this slowed their diffusion. In the case of doxorubicin, which is well known to undergo surface absorption, we saw that happening, so that slowed the diffusion. But when we added protein crowders, the protein crowders could compete with the small molecule for binding to the surface, and therefore that changed the rate of diffusion of the small molecules. And in the case of this drug molecule here, we found that this one had diffusion slowed because of aggregation of the molecules, which could be solubilized by a protein crowder, and therefore increase its diffusion rate. So I don't have time to tell you how we got to finding all that for all those 4 molecules. I'm just going to show you the data for fluorescein to give you an idea of what we saw and how we used the experiments and the computation to tease out what was going on. So fluorescein we first looked at in different solvents. There was no effect on its diffusion rate in these different environments. And one could see in confocal microscopy there was no aggregation. But when we added proteins, again, there was no aggregation, but there were different effects when we looked at the recovery curves. And when we looked at the measured diffusion coefficients, we can see that as you increase protein concentration, there's a big drop with very small amounts of added protein. And then this is the greatest drop is seen for hen egg white lysozyme, then bovine serum albumin, and in fact for myoglobin it drops and then increases. So we were initially surprised to see such effects at such low concentrations of protein, but this showed that this was really an effect of the interactions that the protein could make with the molecule. We next looked at the surface interactions, and here what we see is this is the same plot, so this is the diffusion of the fluorescein, and here's the effect of the lysozyme. If we take the surface and pre-coat it with myoglobin and then add the lysozyme, we get this green plot up here, and you can see that the drop in the diffusion rate is much, much smaller. And similarly, you have a similar effect if you do the same thing with bovine serum albumin. And in fact, in that case, you can also see by labeling the BSA that it actually has a similar diffusion rate to the fluorescein. So that really that these two, at this point when myoglobin is covering the surface, are diffusing, and then the rate is going down due to the effect of adding the protein, excluded volume, interactions, and so on. And in fact, those can be reproduced in Brownian dynamic simulations. We can actually see that there's adhesion on the surface in this liquid droplet by looking here at the where we see the protein. You can see a very sharp peak for the hen egg white lysozyme, a little bit less sharp one for the bovine serum albumin, and when we have the myoglobin surface there pre-treated, we lose that completely. And in the case again of the quinacrine, we can also see that it's localized when we have the hen egg white lysozyme there. So the fluorescein is really attaching transiently to proteins that absorb to the glass surface, and in particular to the hen egg white lysozyme. And so we looked at this with simulations by simulating how the different proteins can interact with a silica-like surface. And you can see with a function of time, you get absorption of molecules to the surface. And we can see that this is greater for the myoglobin and the hen egg white lysozyme. But this is similar for these two proteins. And what is really different for fluorescein is that it has stronger interactions with the hen egg white lysozyme here in green than the other two proteins, which we can see in this radial distribution function showing a peak of interactions at a certain distance that corresponds to the contact distance between the molecule and the protein. And this is perhaps not surprising. Fluorescein has got a negative charge. Hen egg white lysozyme has a positive charge. And here I'm showing electrostatic potentials of these molecules. So blue is positive, red is negative. So you see this is very positive and can interact predominantly in these regions you might see that are sort of green here. But fluorescein can also interact, but more weakly, with these other two proteins, which you can see also have positive patches on them. And that corresponds to where we see the molecule in the simulations. So we can understand why then hen egg white lysozyme has a different effect from the other proteins here. Okay, so this is how we worked out basically what was going on with the fluorescein case. And by similar sort of use of both the simulations and the different experiments, we were able to work out what was going on with these other molecules. And we can really see overall that one has to consider the physical-chemical properties of the individual small molecules as well as the protein crowders and the surfaces to understand this complicated balance that we have here. Okay, I'm running out of time, am I? Okay. Okay, so I want to then just to get to the last topic. There was a little bit of problems with technical stuff. So I'll just tell you a little bit about what we're doing on drug excipient, polymer excipient interactions. So the aim here is to understand how we can formulate poorly water-soluble drugs. And as you probably know, Ape Sangamar has developed a method of making amorphous solid, salt-solid dispersions of drugs and excipient polymers to overcome the aggregation problems of such types of drugs. And we wanted to know how we can really choose a suitable combination of drugs and salts and polymers without simply just doing a systematic experimental screen. Could we use computations to help us work out what to do? And in order to do that, we first really need to understand what's behind these interactions between the